Good morning. So I'll be talking about two different recent epidemiological studies from Faso and also about previous data that suggests that school children are important to malaria transmission. So the studies I'll be describing, they were conceived by Chris Drakely at the London School and Tan Balsama in Nijmegen, and they include infectivity studies led by Alfred Chono and entomological surveys led by Musa Gualbergo, and they were funded by the Gates Foundation. And the first question we need to ask is really what are the factors that influence contribution to transmission? And there are two key factors of phenotype. So gametocyte levels, so density of transmission stage, and the number of transmission opportunities. So there are several studies that have shown that there's a positive relationship between the density of gametocytes and human infectiousness to mosquitoes over the entire range of densities we see in natural infections. And this figure is just illustrating this. It includes data from a study that recruited gametocyte positive individuals by microscopy, and the authors performed membrane feeding assays where mosquitoes are allowed to feed on venous blood through a membrane and then tested for infection 1 week later. And the results of these assays are presented in the y-axis. In the y-axis, and it corresponds to proportional mosquitoes acquiring infection in different membrane feeding experiments. And then in the x-axis we have the density of gametocytes, and the line in the green area, they represent just the output of a statistical model that was used to explain this relationship. But what's important here is just to see that there is a positive nonlinear relationship between gametocyte levels and human infectivity, and it has been seen in several other transmission studies. So the second factor or phenotype is transmission opportunity. So what I mean by this is really the number of mosquito bites an infected individual receives during the infectious period. So if we imagine two individuals of similar gametocyte levels, the individual that receives more mosquito bites is more likely to cause secondary mosquito infections, so is more likely to contribute to transmission. So if you want to estimate a contribution to transmission of individuals, of group of individuals, we really need to quantify these two phenotypes, so gametocyte levels, or quantify infectivity directly with membrane feeding assays or skin feeding assays, and also quantify mosquito exposure. I'll talk briefly about previous data. So if you look at epidemiological data and clinical data, they suggest that children are more likely to carry detectable levels of gametocytes compared to adults. And this figure is illustrating this, includes data from a large meta-analysis that included more than 100 clinical trials. The y-axis is the probability of carrying gametocytes at enrollment. We have in the x-axis age and two different graphs for the African and studies performed in Asia. And we can see that this probability peaks in children in Africa, and then reduces with age, and peaks a bit later in the aging studies. There are also a few studies that quantified infectivity directly using membrane feeding assays or skin feeding assays that allow to see how infectious is an infected individual. And I try to summarize these studies in this graph. So in the y-axis we have prevalence of infectiousness, and infectiousness here is the capacity to infect at least one mosquito in these feeding experiments. And then in the x-axis we have different surveys, and the different colors represent different age groups. And the main message here is really that school children are consistently more often infectious than adults, and you can see that in the different surveys. In our study, our objective was to quantify both age-specific infectiousness prevalence and also mosquito exposure in the same setting, and try to use these two type of data to estimate age-specific contribution to transmission. So we performed infectivity surveys in Burkina Faso in 2 villages during the dry and wet season. We randomly selected participants, so regardless of parasite level, parasite status, and performed membrane feeding assays. So collected venous blood and let mosquitoes feed on the blood, and 1 week later we tested for infection. And just before talking about the infectivity results, I'll just briefly mention the parasitological data we have in this graph. In the y-axis, we have the prevalence of parasites tested by HNS qPCR and prevalence of gametocytes tested by NASPA. So 2 sensitive methods, and in the x-axis, the different age groups for the 2 different surveys, so during the dry wet season. And we can see that, especially in the dry season, the prevalence of parasites and gametocytes is higher in the school children. We use this also these molecular methods to quantify the density of parasites, and this is what's shown in these 3 different graphs for 3 different age groups. So children younger than 5 years, school children and adults, and the x-axis is the parasite density in log scale, and the y-axis is the distribution, is the percentage of the population of different parasite densities. And then we can see that there's a shift to the left in the distribution of parasite densities in adults compared to children. The red line is the median. And if we look at the gametocyte level distribution, we see a similar pattern. So children having higher gametocyte levels compared to adults, although the difference is not that obvious from this figure. Right, but the main objective of this study was really to look at the infectivity outcome. So this is shown in this graph, have the infectiousness prevalence, so again, a capacity to infect at least 1 mosquito in feeding assays, in this case a membrane feeding assay. And in the x-axis, different age groups, and you can see that school children and children younger than 5 years, they were more often infectious than adults. And you can use this data together with demographic data, and this pie chart represents age structure in sub-Saharan Africa. And we can use this data to then estimate the proportion of mosquito infections that originate from different age groups. So just using the infectivity data and the demographic data, but ignoring any difference in mosquito exposure, we estimate that approximately 50% of mosquito infections in that area might have come from school children. But as I said in the beginning, the objective was really to use mosquito exposure data to have a more accurate estimation of contribution to transmission of different age groups. So we collected mosquitoes in one of the study villages, Balongan. We performed 3 different surveys. At the end of transmission season 2013, we collected mosquitoes from 40 houses, and then in the following transmission season, when we collected from 20 houses. So we collected mosquitoes for 5 to 7 days, immediately transferred mosquitoes to the lab, and then dissected blood-fed mosquitoes, separated abdomen with the blood meal, and then we later tested using a multiplex PCR assay that has 9 human microsatellite markers and 1 gender marker. And we also collected samples from the people living in those houses, so and used the same assay, and then we were able to generate a genetic profile for the blood meals and for the humans, and then identify the blood meal sources, so basically who the mosquitoes were biting. We also used the head and thorax to assess malaria infection in mosquito speciation, but I'm not presenting this data today. So we tested more than 1,000 mosquitoes, had 4 possible outcomes, and this is what's showing this graph. In the y-axis, you have the proportion of blood meals in different surveys with the different outcomes. So you can see that between 60 and 70% of the mosquitoes were able to match the genetic profile in the blood meal to someone living in the house. So we were able to identify the blood meal source. For between 10 to 15%, we actually able to amplify human DNA in the blood meal, but we do not match to someone in the house, so mosquito probably fed outside. Similar proportion had multiple blood meals, so we could see that because there are more than 2 alleles in several markers. And around 10% of the mosquitoes, we actually not able to amplify human DNA. So for the next slides, I'll be talking about these green mosquitoes, so the ones we were able to match. So the first question we had was, what's the distribution of mosquito bites in a population? So in the y-axis, we have the proportion of the study population receiving different numbers of mosquito bites or matched to different number of mosquito blood meals. And in the x-axis, you have the number of mosquito blood meals matched to each individual. And data from the peak transmission season is presented by the orange bars, and the data from the other surveys are presented by circles. And so, for example, 30% of the population did not receive mosquito bites or were not matched to mosquito blood meals at the peak of transmission season, and this proportion was higher in the other surveys, so 60% and almost 80% at the end of transmission season. And one thing that's really clear from this graph is that few individuals in each survey have much higher number of matched blood meals compared to the rest of the population. So there's a lot of heterogeneity, and we wanted to estimate the proportion of blood meals or mosquito bites linked to those individuals that received most bites. So in the x-axis, we sorted individuals by number of blood meals matched to them. So the ones that received most bites are in the left of the graph here. And the y-axis represents the cumulative proportion of matched blood meals. So for example, 20% of the population here, the vertical line, received around 75% of mosquito bites in the peak transmission season and almost 95% in the end of transmission season. We use this data then to look at the effect of age on exposure. That was our main question. And we can see in the y-axis the number of mosquito bites or blood meals matched to different study participants. The x-axis is age, and the different circles represent different individuals, and in different colors, different surveys. And the purple line and the purple area, they are just output of a model that explains this relationship. And what's important is that adults, they received 20 times more bites than children aged less than 5 years, and 3 times more than school children. So they're more exposed, and this is really relevant if you want to estimate contribution to transmission. So just to illustrate this, if you think about the calculation I showed before, where we use infectivity data in the same area, we use age structure of the population. We estimated that, but ignoring mosquito exposure differences, we estimated that school children contributed most transmission. Now that we actually can incorporate the mosquito exposure data, it seems that both adults and children are really important for transmission, and children aged less than 5 years in this area, they seem to contribute very little to transmission because they're much less exposed. They represent a smaller fraction of the population. So it was really relevant to actually adjust for this. So just to summarize, we saw in this area that school children they carry parasites and gametocytes more often than adults. They have higher densities, they're more infectious, but adults are more exposed to mosquitoes, and actually this just balances their contribution. So both school children and adults contribute most transmission in this area, and children aged less than 5 years contribute a bit less. And that's all.
Thank you, Bronner. And again, we'll hold questions until